Tantor Audio, a division of Recorded Books, presents The History of Life, a Very Short Introduction, by Michael J. Benton, narrated by Peter Larkin.
Introduction. The age of reptiles ended because it had gone on long enough, and it was all a mistake in the first place. Will Cuppy, How to Become Extinct, 1941. It is hard to make sense of the history of life on Earth. A mass of strange and extraordinary animals and plants perhaps flits before our eyes when we think of prehistory. Neanderthal man, mammoths, dinosaurs, ammonites, trilobites, and, of course, a time when there was no life at all, or at least merely microscopic beasts of extreme simplicity floating in the primeval ocean. These impressions come from many sources. Children today are weaned on dinosaur books, and the images of living, breathing dinosaurs are everywhere, in movies and television documentaries. Then, too, as children, many people have gone to coastal cliffs or quarries and collected their own fossil ammonites or trilobites. These common fossils, as well as many much more spectacular and beautiful examples, such as petrifactions of exquisite fishes showing all their scales, still shiny after millions of years, may be seen in fossil shops or in lavish photographs in coffee table books and on the web. Most people are aware that dinosaurs, despite their ubiquity in modern culture, lived a long time before the first humans, and there were untold spans of time before the dinosaurs existed that were populated by ever more unusual and strange animals and plants. How are we to make sense of all this? Fossils. The keys to understanding the history of life are fossils. Fossils are the remains of plants, animals, or microbes that once existed. Fossils may be petrifactions, which means literally turned into rock, and these are some of the commonest examples. Petrified fossils may be of two kinds. First, those that are literally turned to rock, and where none of the original organism remains. The leaf or tree trunk or shell or worm has completely disappeared, and the cavity left behind has been replaced by grains of sand or mud, and more often by minerals in solution that have flowed through the spaces in the surrounding rock, and have then infiltrated the space and crystallized. The second and commoner kind of petrifaction still retains some of the original material of the animal, perhaps the calcium carbonate that made up the shell, or some cuticle or carbonized relic of the plant. Rock grains or minerals then merely fill the cavities. So, many people might be surprised to realize that common fossils, such as a 400-million-year-old trilobite or a 200-million-year-old ammonite, are actually largely made from the original calcium carbonate of their external skeleton or shell, as in life. Similarly, by far the majority of dinosaur bones are still made of the original calcium phosphate, apatite, the main mineralized constituent of bone then and today. If you look closely at the outer surface of these fossils, perhaps with a magnifying glass, you can see extremely fine features, such as pimples and growth lines on the trilobite carapace, original multicolored mother-of-pearl on the ammonite shell, and muscle scars or tooth marks on the surface of the dinosaur bone. If the fossil shells or bones are cut across and examined under the microscope, all the original growth layers and internal structures are still there. So a section cut through dinosaur bone looks just as fresh today as a section through a modern bone. Every plant or animal that has ever lived has not turned into a fossil. Indeed, if this were the case, the surface of the earth would be covered in avalanches of fossils everywhere, great mounds of dinosaur bones, trilobites, giant coal forest trees, ammonites, and the like, probably extending to the moon. No one knows what proportion of life has ended up fossilized, but it is clearly a tiny fraction, much less than 1%. Plants or animals must at least have hard parts, such as a skeleton, a shell, or a toughened woody trunk to be readily preservable. Even so, the majority of animal carcasses and dead plants enter the food chain almost immediately, being scavenged by animals or decomposed by bacteria. Dead organisms can only turn into fossils if sedimentation is happening. That is, sand or mud are being dumped on top of the remains, perhaps on the floor of a deep lake, under a sandbar in a river, or deep in the ocean, below the zone that is constantly churned up by currents and tides. Worms and feathered dinosaurs. Exceptional preservation. Other fossils may be preserved in slightly unusual conditions that may, on occasion, provide unique and unexpected insights into ancient life, so-called exceptional preservation. Exceptionally preserved fossils may show soft structures such as flesh, eyes, stomach contents, feathers, hair, and the like. Sites of exceptional fossil preservation are sometimes called windows on the life of the past. They allow paleontologists, the scientists who study fossils, to see a snapshot of everything that existed at particular times and in particular places. These at least allow paleontologists to see the soft-bodied worms, jellyfish, and other creatures that are rarely preserved in normal circumstances. The Burgess Shale in Canada is one of the most famous of these sites of exceptional preservation. These rocks are 505 million years old, so they document some of the oldest animals. Without the Burgess Shale, and similar sites of about the same age in Greenland and China, paleontologists would know only about shelled and skeletonized organisms such as brachiopods, lampshells, trilobites, and sponges. The Burgess Shale has increased our knowledge of life in the Cambrian manifold. It has revealed whole clans of worm-like creatures, some related to modern swimming and burrowing worms, others seemingly unique and hard to link to modern animals. The Burgess Shale also shows the feathery legs and gills of the trilobites, their mouths, guts, and sense organs, and it reveals strange tadpole-like swimming animals that have primitive backbones, and so are close to our own ancestry. Equally famous are the sites of exceptional preservation in Liaoning Province in northeast China. These date back to 125 million years ago, and they have produced spectacular fossils of birds and dinosaurs with feathers and internal organs, mammals with hair, fishes with gills and guts, and any number of worms, jellyfish, and other soft-bodied denizens of those ancient Chinese lakes. There are dozens of other such sites of exceptional preservation scattered pretty randomly through time and space. But why do they exist? And how are the soft structures preserved? Most of these sites come from times and places where oxygen was limited. Deep lakes and deep oceans sometimes lose the normal oxygen content of the waters, if, for example, there is a dramatic growth of algae and other floating plants at the surface, a so-called algal bloom. These occur in warm conditions, and the lakes and oceans may become temporarily stagnant. The stagnation of the waters may itself kill swimming creatures and beasts that crawl around on the bottom muds. The lack of oxygen can also mean that the normal scavenging creatures cannot survive, and the carcasses do not have all their flesh stripped. Experiments show that in oxygen-poor or anoxic conditions, soft tissues, even muscles, guts, and eyeballs, can be invaded by minerals that come from the body fluids of the animals, or from the surrounding sediments. These are typically flash mineralizing processes, where the fibers of a muscle, or the complex tissues of a gill or a stomach, are invaded and replaced within hours or days at most. Once mineralized, the replicas of soft tissues can then survive to the present day. Living blimps? Quality of the record. Like most paleontologists, I sometimes sit bolt upright in bed at night and worry whether the fossil record is informative or not. Charles Darwin wrote about the imperfection of the geological record, and he was well aware that most organisms are never fossilized, and so paleontologists miss so much of ancient life. The question, though, is how much is missing? Is it 50%? Or 90%? Or 99.99999%? This can never be determined, of course. A more sensible question might be, how adequate is the fossil record? Paleontologists have speculated that there might be whole sectors of extinct life that we know nothing about. What if there were a diverse class of floating animals that were constructed of extremely lightweight materials and provided with great air bladders that filled with gases lighter than air? These creatures might have been many meters long, perhaps as large as dirigible aircraft, sometimes called blimps during the Second World War. These blimp beasts could well have dominated the Earth if they were so large, and yet they might have entirely escaped fossilization. Their bodily tissues might have been so lightweight that they rotted away when they died. Their gas bladders would clearly burst and disappear during decay. Living in the air, in any case, means their carcasses might have generally fallen onto the surface of the Earth, and so they might not often have been covered with sediment in any case. Paleontologists have no way of detecting such hypothetical extinct beasts. Other soft-bodied creatures can be assumed to have existed, though. For example, there are many phyla or major groups of worm-like creatures today: nematodes, platyhelminths, gastrotrichs, sipunculids, and others that have no known fossil representatives. And yet, because they exist today, and because we can establish their evolutionary relationships to other organisms with shells or skeletons, we know the length of their missing fossil record. If a soft-bodied worm group is the closest relative of another wormy creature with a shell, both groups must have existed for the same length of time. Their common ancestor must have lived at a particular time, and the fossil record of the shelled group establishes a minimum age for both groups. The known missing record of the soft-bodied group is called a ghost range, a part of the missing fossil record we can predict with some certainty. What do the sites of exceptional preservation tell us? If they preserve more or less everything that lived at the time, soft and hard-bodied, they can be used as a yardstick against which to test the normal fossil record. It seems that the ancient exceptional sites, such as the Burgess Shales, tell us more about unknown groups than the more recent ones, such as the Liaoning beds in China. In fact, the soft-bodied organisms from Liaoning, worms, jellyfish, insects, and the like, are all entirely predictable from other known fossils and from ghost ranges. Paleontologists have been pretty assiduous in retrieving fossils. As time goes on, it now seems to take much more effort than it took a century ago to find something new. Indeed, not much has changed in our knowledge of the fossil record since the time of Darwin. In the 1850s, paleontologists knew about trilobites and ammonites, fossil fishes, dinosaurs, and fossil mammals. They did not know anything about the first life from the Precambrian, nor did they know much about human evolution. But the fact that neither trilobites nor humans have been found in the age of the dinosaurs, nor have any other fossils been found in seriously unexpected places, suggests that the record is known more or less well. Our work now is merely to flesh out the details. But that still says nothing about the giant blimps. Molecules and the history of life. It might seem unexpected to introduce molecular biology at this point, but just as historians have parallel sets of evidence from artifacts and from written records, so too do students of the history of life. Until the 1960s, there were only fossils. After that, there were also molecules, even though most paleontologists at the time probably did not appreciate it. In an extraordinary paper published in 1962 by Emil Zuckerkandl and Linus Pauling in a rather obscure conference volume, the molecular clock was born. Molecular biology had arisen 10 years earlier, when in 1953, James Watson and Francis Crick announced the structure of deoxyribose nucleic acid, DNA, the chemical that makes up genes and is the basis of the genetic code. By 1963, several proteins, such as hemoglobin, the protein that carries oxygen in the blood and makes it red, had been sequenced. That is, the detailed structure had been determined, and the new breed of molecular biologists had noted something extraordinary. The proteins of different species of animal were not identical. and their structures differed more between distantly related species. In other words, the hemoglobin molecules of humans and chimpanzees were identical, but the hemoglobin of a shark was very different. Zucker, Kandel, and Pauling took the brave leap of suggesting, on rather limited evidence then, that the amount of difference was proportional to time. The negligible difference between the hemoglobins of humans and chimpanzees showed these two species had diverged only a short time ago, geologically speaking, whereas the 79% difference between human and shark hemoglobin pointed to a divergence 400 million years ago or more. In the 1960s, protein sequencing was a laborious process, and the new data came slowly. But by 1967, the hemoglobin of the great apes was known sufficiently that the first attempt was made to produce an evolutionary tree. The science of molecular phylogenetics was born. Vincent Sarek and Alan Wilson, in a 3-page paper in the American journal Science, plotted the relationships of humans and apes and showed that our nearest relative was the chimpanzee, then the gorilla, and then the orangutan. This was not so unexpected, and it agreed with the pattern of relationships established from studies of anatomy. The shocking part of the paper was that the molecular clock said humans and chimps had diverged only 5 million years ago. Paleontologists were variously bemused and horrified. Most dismissed the new technique. After all, if it produced such ludicrous results, it was clearly not working. Everyone knew that humans and chimpanzees had split some 15 to 20 million years ago, based on studies of proconsul and other early human-like fossils from the Miocene of Africa. Others took the method seriously, but were equally unhappy about the result. As the protein datasets grew, more mammals were added to the tree, and the branching dates seemed quite reasonable for most other groups. This increased the nervousness of the paleontologists, who then faced a conundrum. Do we accept the new molecular date, or insist on the established fossil evidence? Slowly they came to realize the molecular date was probably right. Closer study of the fossils showed that they had been overinterpreted. The supposedly human characters of proconsul and its kin were not really human at all. This fossil was related to the common ancestors of humans and the African apes, and so said nothing about the true timing of divergence. Since the 1970s, new finds in Africa have shown that the divergence date between humans and chimps must be at least 6 to 7 million years ago. Now molecular biologists interested in the tree of life, the great pattern of relationships linking all species, use DNA sequences. Protein sequencing is slow, and the evidence limited. DNA, the genetic code, offers much more information, and new techniques developed in the 1980s have made sequencing almost automatic. Computers can also crunch enormous masses of data these days, so sequencers are happy to run lengthy segments of the genetic code, consisting of many genes, and for dozens or even hundreds of species, to produce patterns of relationships for specific groups or for large sectors of life. It is possible to assess the genome of, say, 20 species of lizards and draw up a tree that documents evolution over a span of perhaps 10 million years. Equally, the analyst can select, say, 20 species across all of life: a human, a shark, a mollusk, a tree, a fern, a bacterium, and find a tree of relationships that extends deeply back in time. But where do the fossils fit into all this? Cladistics. I remember when I attended my first scientific meeting, as an undergraduate, a session of the Society for Vertebrate Paleontology and Comparative Anatomy at University College London in 1976, I wondered if I would ever go back. As I looked on nervously, the big beasts of the subject were bickering and squabbling appallingly over something called cladistics. I'd heard nothing about this. It wasn't taught then as part of my degree. One person would assert with fervor that everyone should adopt this new technique. Another would say it was all nonsense, even a Marxist plot to overthrow the scientific method. I stumbled back to the train, wondering whether my decision to become a professional paleontologist was mistaken. Were they all mad? On reading around, I discovered that cladistics had been promulgated by a German entomologist, Willi Hennig. He had written about the technique in the 1950s, but it had only really attracted attention when the book was translated into English and reissued in 1966. But from 1966 to 1980, only a rather small group of true believers espoused the method, and it had not in any way become mainstream. Hennig argued passionately that systematists, the biologists and paleontologists who were interested in species and the tree of life, should be more objective in their methods. Until Hennig's time, systematists had attempted to draw up trees of relationships based on a judicious sifting of the character evidence. A biological character is any observable feature of an organism: possession of feathers, possession of 4 fingers, iridescent blue feathers on top of the head, multiple flower heads on each stem. And systematists had long understood that if 2 organisms share a character, they might well be related. The problem was always convergence, the well-known observation that unrelated organisms might evolve similar features independently. Insects, birds, and bats have wings, but no one ever suggested that this was sufficient evidence to group these organisms together as close relatives. In detail, their wings are anatomically quite different in structure, and so they evolved them independently, but for the same purpose. But how were systematists to distinguish convergence from truly shared, evolutionarily identical characters? This was Hennig's point. Objective techniques were required to distinguish truly shared characters from convergences, but also to distinguish inherited primitive characters from those that truly marked a particular branching point. So, while it is true that humans and chimpanzees share the character hand with 5 fingers, and this is not a convergence, the character is not helpful at the level of the branching point between the 2 species. In fact, all land-living vertebrates basically have a 5-fingered hand: lizards, crocodiles, dinosaurs, rats, bats, whales, and so on. Hennig had identified the critical point, that anatomical characters had to be evolutionarily unique, not convergent, and they had to be assessed at the correct level in the tree before they could be considered useful. He termed such characters synapomorphies, sometimes rendered in English as shared derived characters. Hennig's writing in any language is heavy-going, and he liked inventing long words, neither of which helped gain him converts. Hennig's concept of a synapomorphy is more or less the same as the classic notion of a homology, that is, any structure that shares a common fundamental pattern because of common ancestry, such as the human arm, the wing of a bat, and the paddle of a whale. These limbs may have different functions today, but they all share the same bones and muscles inside, and we now know they evolved from the ancestral front limb of the first mammal. Since the 1970s, systematists have increasingly switched to using cladistics in their work. After all, there was no alternative. The older techniques were really inspired guesswork. Acceptance came largely for a reason Hennig could not have predicted, namely the growth in power and ease of use of computers. The secret to cladistics is the character matrix, a listing of all the species of interest and codings of their characters, 1 for presence, 0 for absence. Multiple cross-checking over the matrix and repeated runs of the analysis provided statistical methods of assessing which tree or trees explained the data best, and the probability that synapomorphies were correctly identified or not. In practice, there have been many problems, but cladistic methods are ubiquitous, and repeat analyses by different analysts allow published trees to be tested and confirmed or rejected. The Great Leap Forward. Paleontologists are aware that their field has transformed itself immeasurably since the 1960s, but public attention has focused elsewhere: the space race, genetic engineering, computer technology, nanoscience, global change. But cladistics and molecular phylogeny have introduced new rigor into the field of drawing up evolutionary trees. whereas in the 1950s and 1960s, a paleontologist did his or her best to make a tree by joining the dots, linking similar-looking beasts through time. Today there are many independently derived trees of the evolution of different groups, some based on different genes, others on different combinations of fossil and recent data on anatomy. But do they agree? The astonishing discovery is that molecular and paleontological trees agree with each other more often than not. The two approaches are pretty well independent, so it is possible then to compare, say, a tree based on molecular sequences of modern rodents with a tree constructed by measuring the teeth and other anatomical features of living and extinct species. Inevitably, everyone hears about the cases where the results disagree. In the early days of molecular sequencing, some bizarre results emerged, but the methods were young and mistakes were easy to make. Such bizarre results are rare now. In some cases, paleontologists have humbly accepted that they have been entirely unable to resolve certain parts of the evolutionary tree, and the molecules give an unequivocal answer straight away. In other cases, there is no resolution yet, and more work is required. Some parts of the great tree of life may remain forever mysterious, perhaps because rates of evolution were so fast that characters did not accumulate, or the branching points are so ancient that subsequent evolution has obliterated the clues to relationship. The third methodological or technological advance has been in dating the rocks. Since the 1960s, the accuracy of dating has improved greatly, and sequences of rocks and sequences of events can be compared more accurately than before. But we can look at that later. Let's begin the story. Chapter 1. The Origin of Life. As a general rule, then, all testations grow by spontaneous generation in mud, differing from one another according to the differences of the material. Oysters growing in slime, and cockles and the other testations above mentioned on sandy bottoms, and in the hollows of the rocks the ascidian and the barnacle and common sorts, such as the limpet and the nerites. Aristotle, History of Animals. From the earliest days, people have wondered about the origins of life. The ancient Greeks and Romans considered the topic and had many ideas. Most, like Aristotle, 384 to 322 BC, focused on the idea of spontaneous generation, a process that they believed happened today, and that had presumably happened when life first arose. As Aristotle wrote above, he believed that marine shellfish all arose spontaneously from the mud, sand, and slime on the sea bed and among the coastal rocks. He made similar assumptions about other forms of life. Moths arose from woolen garments. Garden insects arose from the spring dew or from decaying wood. And many fishes arose from froth on the surface of the ocean. Such views held sway until the 19th century. Louis Pasteur, 1822–95, famously showed conclusively that life could not arise spontaneously. He repeated experiments that had been performed before, but took great pains to exclude all possibility of contamination. Earlier workers had gone through the process of boiling a broth of water and hay in sealed flasks so that anything living in the water or the air within the flasks would be killed. But despite these precautions, they still found microscopic organisms living in the water, and Pasteur argued that the germs entered the vessels when they were being cooled in a mercury trough. So he repeated the experiments, sterilizing the glassware and the water in the flasks, but ensuring also that laboratory air could not enter the cooling mixtures. With the air excluded, nothing living was detected in the boiled water, even many months later. The Age of the Earth. The death of spontaneous generation was not the only problem for scientists interested in studying the origin of life about 1900. They also had no truly ancient fossils to work with, and no real idea of the age of the Earth, nor of the major events that might have preceded the origin of life. There was a widely held view that the Earth was something like a huge ball of iron. Iron is one of the commonest elements that had once been molten and had been cooling down. Indeed, the eminent late Victorian physicist William Thomson, later Lord Kelvin, 1824 to 1907, used this assumption and his knowledge of thermodynamics to speculate that the Earth formed only 20 to 40 million years ago.
Sample complete. Ready to continue?